Good evening, ladies. Please be seated. And gentlemen. But you must agree it's a ladies' event. Prof. Deborah Meyer, who's the Executive Dean, Faculty of Science. Prof. Michelle van der Bank, who's our most important person here tonight. Dr. Monica Mwale, who's our respondent. Senior leaders of the university, members of Senate and other academics, distinguished guests. Ladies and gentlemen, Sanabonani, goeienaand, good evening, and Tubela. I'm standing in for the Vice-Chancellor, Professor Chalitsi Marwalo, who unfortunately can't be here. He sent his apologies and also he asked me to convey his congratulations to Michelle. So my name is Kinta Burger and I'm the registrar, but I am the VC for tonight. So ladies and gentlemen, it's indeed a great honor and special privilege for me to welcome you to the Professorial Inaugural Lecture of Professor Michelle van der Bank. And as I do so, I wish to express a warm welcome to her loved ones, special guests and other colleagues. This is a proud, joyful and landmark moment for all of you and for Professor van der Bank in particular and of course for us here at UJ and higher education in South Africa and beyond. The inauguration of professors is a public ceremony in which newly appointed professors are inducted into and deliver their inaugural addresses. And this ceremony serves multiple purposes. Firstly, it is an expression of welcome and an entry for new professors joining the circle of colleagues who are already professors. And secondly, it provides a platform for the professors to display their expertise in the discipline and showcase their research. It is a celebration of the contributions to the discipline and ultimately the impact on society. Professors provide the university with its identity, character and academic integrity. And it stands out also as a moment of pride for the incumbent, the family, fellow scholars, the university and ultimately society. Today, ladies and gentlemen, we gather to witness the entry of Professor van der Bank to the illustrious community of scholars at the university. The inaugural lecture is a rite of passage following the confirmation of the appointment of the person as professor. And in the case of Michelle, that happened in 2016 when she was promoted to full professor. So this evening we will listen to Professor van der Bank as one further step in the journey of being a professor. This is a journey which does not culminate once this lecture has been given. It is a self-reflective pause in the journey of the professor, with a promise of more to enrich our minds and simultaneously contributing to the rich intellectual body of work in the discipline. The power of the inaugural address is when the expertise is showcased beyond the corridors of the university and reverberates with society. Michelle, we are looking forward to your address, and I now ask the Executive Dean to come and introduce you. Thank you, ladies and gentlemen.
Good evening, ladies and gentlemen. It's a privilege for me to introduce Michelle van der Bank, and I think I'm going to remind her of the good old days by just mentioning we were students together. I think she was maybe a year or two ahead of me, so she was a demi. So it's good to be part of something that is a long time in coming. Michelle van der Bank completed a BSc and BSc Honours Cum Laude in Zoology, a BSc Honours Cum Laude in Biochemistry at the Rand Afrikaans University, which is now University of Johannesburg. She received a PhD in Botany from the same institution in 1999. In 1996, Michelle was appointed as a lecturer in Botany, where she was promoted to full professor in 2016 at UJ. She is currently the director of the African Center for DNA Barcoding. Her research group uses molecular phylogenetics, comparative analysis, and intensive fieldwork to address questions relevant to biodiversity science in Africa. She also leads important research into the authenticity and conservation status of traded plant products, including medicinal plant species and timber trees. Additionally, Michelle has played a leading role in building capacity needed for African researchers to participate in the International Barcode of Life, or IBOL project, the largest endeavor ever undertaken in biodiversity genomics. More than 30 postgraduate students have graduated under her supervision, and she has authored or co-authored more than 80 peer-reviewed publications and has an H-index of 25. She is also currently NRF rated with a C1 rating. Michelle is a founding member of the International Scientific Collaboration Committee of Canada for IBOL, and also one of the project leaders for the Planetary Biodiversity Mission, a project that will provide the means to provide a comprehensive picture of multicellular biodiversity on a global scale. So with that said, I introduce you to Michelle van der Bank.
Hello everybody. I just want to move my computer. Thank you. Thank you, Prof. Kinterberger, Debra Meyer, Monica Mwale. It's actually for me a privilege that the three ladies are here tonight because I think we came a long way. Prof. Berger being my previous dean, now Debra, and me and Monica are starting off working on the IBOL project probably in 2005 together. So thank you very much for the kind words. And thank you to all of you guys being here tonight. My title of my address would be, of course, DNA barcoding. This is my passion. And what we do at the African Center for DNA Barcoding. I will discuss to you the current applications and also our future prospects for the lab. And I want to draw your attention to this little quote here, the IBOL quote, make every species count. Whether it's a small banana or a big gorilla, everything is important for us. Okay, I want to start off my address actually by acknowledging people that has played a big role in my life. And firstly, the University of Johannesburg, specifically the Faculty of Science, and my home department, the Department of Botany and Biotechnology. You guys have been absolutely fantastic. I would never leave this university. It's my house. So thanks very much. And then there was four men that played a very big role in my life, and some of them are here. First, Prof. Ben Eric van Wyk, taking me as a PhD student, shaping me for the future. I'm very proud to be your student, and I've learned a lot, and I continue learning from you. And Prof. Mark Chase from Kew Botanical Gardens, who played a big role also in my PhD studies. Prof. Richard Pinoff, my previous boss from Wits University many years ago, before 1996. And then Paul Heber, who has been an inspiration for all of us in the Barcode project. Then my family, who's very important to me, my husband Herman, my late parents who can't share this day with me, and then my family that's here, my sister Teresa, Gerard, Andre, Madeleine, my mother-in-law, and Sarina. Thanks very much for attending this with me. Then very important is also my collaborations, because they shape your future, change your ideas on how you address your research questions. So I've listed a few here or showed a few faces here of people that's been really important, and some of them were actually my ex-students that has become lecturers in their own right, and working very closely with them. And then last but not the least, my students, which I can see here, all dressed in your African outfits for the African Center for DNA Barcoding. You make me proud. You are an inspiration to me, and I think the center is what it is because of you guys and the effort that you put into the work at the center. I know I can be tough, but you guys are just wonderful. So thank you very much. So when we get back to my address, I've actually structured the talk into 3 parts. I would first say where we stand with biodiversity science, possible solutions to the problems in biodiversity science, and then I will focus on the research that we're doing at the African Center for DNA Barcoding. So I think the first question when we look at biodiversity science is how many species is on Earth? We don't know. Since Linnaeus, there has been 1.7 million animals and plants being described. And even though this is impressive, there's actually an estimated 10 to 100 million species alive today. So we have a big job ahead of us. And if we work this out, it would take us, if we use conventional methods, another 1,500 to 15,000 years to describe all the species. And you all know that species disappear every day. So we have a big job ahead of us, and my students know this, so, and Monica would agree on this. So a second challenge when we look at biodiversity science is also to recall the names once they've been described. I mean, in your mind or in any taxonomic person, a taxonomist's mind, you probably can recognize 1,000 species and know the diagnostic characters. Maybe my supervisor, Prof. Ben Eric, might know more, but it's quite a challenge to know 1,000 species. So we have a big problem there. And the biggest problem is probably that we are losing taxonomists, especially in South Africa, but it's also a global problem that they are not replaced. People are retiring, and we don't get young people into this field. So we must make it exciting for young people to go into this field. And we're trying to do it with DNA barcoding to attract people to this field. So where did it all started? It started, this is the first barcoding paper that actually was written. And this is Dan Jansen that you can see here, very enthusiastic person. And he actually heard about this DNA barcoding that Paul Herbert is advocating, and he arrived at a conference with a suitcase full of skipper butterflies that occurs in Costa Rica, and according to him, they all look the same, and if you look at this slide here, you will agree. There's a little bit of color variation, but essentially, they're one species. So, but if you look at the caterpillars of this skipper butterfly, they do differ. You get all these different color patterns on it. But the problem is they're never alive at the same time. You either get this life form or this life form. So he brought 500 specimens to Paul Herbert. They sequenced it. And what they got in the end is all these lineages, 10 different lineages. And essentially that was a species complex with 10 species. So this is what barcoding did. So if we go back to what actually is a barcode, it's basically a very short region in a genome that you use to identify species, or you can also assign unknowns to a species. Essentially, it's about 650 base pairs long. And if you compare it with, for instance, if you go and buy groceries, and this is actually how the whole idea came about. Paul Herbert was in a grocery store, and they were scanning his groceries, and he said, well, if that can work using a universal product code, why can't it work for distinguishing species? So this is how this whole concept started. So for barcoding, we use the 4 nucleotides A, C, T, and G to distinguish between species. And this sounds now very easy that we say the animals have one barcode, CO1, plants have 4 because we don't have a lot of divergence there, and fungi 1. But that took a lot of years of testing different regions to come up with this specific barcode. So although barcoding was first advocate in 2003, the first paper was in 2004, the International Barcode of Life project was only initiated in 2010. And as Prof. Meyer was saying, it's one of the largest biodiversity initiatives ever undertaken. It's a huge project. Here on this slide, you can see all the countries that's currently involved in this project. And when we started the project in 2010, we had a goal of producing 5 million barcodes within the next 5 years. So I think it was August in 2015 when we actually met this target of producing all the barcodes. If I say we, I'm talking obviously of all the collaborators around the world. So our ultimate goal was to have this barcoder where if you're in the field, you just take a leg of an insect, put it in, does everything in this little machine, whoops, the name comes out. Okay, this was the idea. And I remember still the first talk that I gave about, and I was mentioning this barcoder, I had a handwritten letter from a guy who was in Alberton, and his son or his grandson was studying biology. And he wanted to buy one of these barcoders. And I said, oh, well, you'll have to wait a few years. We don't have it yet. But as I will go through the talk, you will see we are there very soon. So this idea is not forgotten, even though a lot of people were saying this is never going to happen. I mean, I was just looking what happened in my career in biochemistry, the equipment that we used 15 years ago. I know I was a student a very long time ago, but 20 years ago, make it 30. But things have changed, things have scaled down. So this for me was always a possibility that we're going to get to this point. And what was very interesting, and the timeline here, we're looking 2013 to 2017, is that in the beginning, everybody was building this database, this reference database that we're going to match our sequences with. So you get a high number of publications here, and it has now decreased. Not so many people are building just reference databases, but what has increased is the socio-economic importance of using DNA barcoding, the applications. This is the important thing. And I'm going to show 3 examples here, and when I talk about what we do at ACDB, you'll see that we have really focused on this the last 4 or 5 years, that aspect. So the first one is monitoring of market samples. And there's this classic example, and I really love this story because it's young kids. It's high school kids that heard about DNA barcoding. They live in New York, so they went out to all these fish shops. They bought some fish, sampled a little bit, and they sent it off to the Canadian Centre for DNA Barcoding. And what they found is that, for instance, white tuna, which is quite expensive fish, was supplemented with Mozambican tilapia, which is quite a cheap fish. So out of their 60 samples that they used, a quarter of those samples were mislabeled. So you can see the huge effect that this could have on the fish industry. And Monica is maybe more involved in this, being a fish person. But what was very interesting here is that you can see 380 TV shows just reporting on this case study. And this was translated 10 languages. It was on TV in 34 countries, so a big issue. But the most important thing was that the FDA actually now adopted DNA barcoding to detect whether the seafood fraud is taking place. And that happened in 2011. So from being a school kid to this is wonderful. And this is just showing you that barcoding can be done by everybody. And we at the ACDB actually are giving courses to young school kids in DNA barcoding, because this is something that excites people, and Ronnie here is quite involved in that. And then another example, for instance, is the illegal bushmeat trade, and this is very relevant to Africa. And again, this example is not from Africa, but this is what we're seeing almost every day in the zoo, especially. It's getting lots of cases in. So this was a case in Brazil where they caught these guys with the meat, and the suspects claim it was pork that they had. Obviously, there's no way that you can say that this is pork or this is antelope or whatever. So, and to enable appropriate law enforcement, you actually need to know what is the species that you're dealing with. So again, they did CO1, the barcoding gene. They sequenced these samples. They blasted against the reference data set, and it was a pampas deer, which is a near-threatened little bambino. And these guys were found guilty, and they were sentenced to prison. So a very good application of where DNA barcoding has worked. Then the illegal trade in endangered species, which is a big problem in South Africa. Our colleagues from the DEA is here, so they can agree with this is really a nightmare for us, because you're dealing with bones, you can't identify it, or cycad pieces, you don't know which species it is. So, and again, we've used DNA barcoding to sort this out. So again, this is another example here. This is a guy that got caught in Australia trying to get through the border from a flight from Bali. And they actually found that he was carrying 39 eggs in this nice little vest that he was wearing. I'm not sure how you can take that chance going into Australia, but anyway, he did that. And then what they did is they sequenced these eggs, and what they found is 6 species of macaw, African grey parrots, and 2 eggs from a salmon-crested cockatoo, which is a CITES 1 species. So again, a very good example of barcoding. In this case, they pleaded guilty, and they were sentenced to 2 years in jail and a $10,000 fine. So now I'm going to move to the third part of the talk, more focusing on African Center for DNA Barcoding and what we do there. This picture is one of the Toyota outreach pictures. Toyota sponsored barcoding. I think it was 5 years from the beginning of barcoding, where every year they actually arranged a trip for 2 weeks around the country, where they fed us, they drove us around, we were collecting specimens, and this is how actually we got very, very quickly specimens for DNA barcoding. So, but our focus at ACDB for the past few years have been on woody plants. And in the beginning, I was very optimistic and enthusiastic. I said, we're going to do the whole of Africa. But obviously, the whole of Africa is big. Collaborations is a bit tricky to find higher than Southern Africa. We're now starting to get collaborations in Central Africa, but it's a big task. So we thought rather we stick to Southern Africa. Once we've done that, we'll start slowly moving up. But anyway, it took us to wonderful places, as you can see here. Now we're jumping. Wonderful places. The Kruger National Park. This was taken in Zambia. You see the guys are tired. But anyway, it was a huge experience. But we traveled many, many kilometers. And in the end, we barcoded about 21,000 plants to get to the point where we are today. And that's roughly about 7,500 species. And about 2,000 genera. So it's quite a number of plants. This is just to show you the collection points where we collected. We also collected animals during this time. again, the different habitats where we collected our specimens. So the first step for us was then to use this data and produce a phylogenetic tree for the trees of Southern Africa. We just used two markers, the MATK and the RBSL, the core barcoding regions for this, but the phylogeny was well resolved and we could use it for several ecological applications, which I would quickly go through with you. And we dated also the tree. So our first study was actually in the Kruger Park, and before we actually looked at plant community dynamics to large mammal disturbance, we actually used the Kruger Park also to predict which would be the best barcode for plants. So ACDB was one of the leaders in deciding which plant barcode should be used. But in this study, you can see the people involved here. The student was Kuiu Yosefo, who's now also appointed at UJ. We did the following. We produced a phylogeny just for the trees and shrubs of the Kruger National Park. We recorded abundance, so where they occur and how many, in 110 plots across the KNP. And we call this herbivore unrestricted areas, plots, so where big herbivores can eat freely, so elephants, giraffes, everything can get in there. And then we also had our restricted herbivore plots, and there was 15 plots that we used, and this was long-term exposures in the park. And we wanted to see what is the impact of herbivores on plant community structure here. So what we found here was quite interesting because you know the big debate about should we cull elephants or not. And this is where it comes in quite a bit. Our idea is that you should not cull them, but I mean, this is up to you to decide. But we found that exclusion of large herbivores actually resulted in a decrease in plant species diversity. So you got less diversity. And also that the plant community response to herbivores exclusion depend on the initial structure of that community. So if you have communities that's initially phylogenetically over-dispersed, they end up being under-dispersed after herbivore exclusion. And the same was true when the communities was initially under-dispersed. Then you get over-dispersion after you exclude the herbivores. So the significance here for the Kruger Park was that under-herbivory might be as damaging to the Kruger Park plant communities as over-herbivory. So if you want to start excluding or culling animals, you have to really very carefully research this and see what would be the outcome. Don't make just hasty decisions. So the next study that we did also looking at herbivory was the importance of mammal browsers in the opening of forests in Africa, and we use spinescence here as a marker for mammal herbivory as an indicator. So again, we use the main data set that we produce using the two barcodes. In this case, we moved higher up in Africa where we got collaborators in Belgium that sampled quite a bit of sampling in the Central Africa. So we had initially about 1,800 tree species into this database, and we used the published phylogeny for bovids on the work of BB et al. In 2013 for this work. And what we found is that spinescence actually evolved at least 55 times in this phylogeny. So the significance of this study was the data suggests that browsers predate fires as agents driving the origin of African savannas. So this was quite an important conclusion that we made. And then a paper by Morin et al. That actually got a lot of attention. And if you look here at this drawing here, it's It was an Australian guy that read the paper, an artist, and he thought, oh, this is cool, plants that grows underground because we call it African forest, underground African forest. And he came up with how he sees the underground forest. But in any case, here we explore the origins of the Suffratex life form, or the so-called underground trees, and we consider whether the species provide a marker for the appearance of humid savannas. So we again use the same phylogeny that we produce using the barcodes, but in this case we added 58 Suffratex taxa, the so-called underground trees, and these plants are just wonderful. I'm going to show you some pictures, but you can see 2 of them here also. So they basically grow underground. The stem, the branches, everything is underground, and then once a year it puts out shoots above ground, it flowers, it fruit, and choom, goes underground again. So absolutely amazing. But what's important here is they normally appears in clades where they the only Suffratex, or the underground tree, and the rest are trees, proper trees. And if you look at their distribution, you get them here in the Sudanan area, only 7 species occurring there. And then in the Zambesian region, you actually get 200 species. So they're quite widely distributed there. Parinari capensis, the most widely distributed species of this. And just to show you what I mean by they fruit and flower. So this is the sister tree species. You can see a proper tree, Gardenia ternifolia. This is the Suffratex. So on ground level, you get the fruits and the flowers. And this happens one season, and then they go down underground again. And we went and collected them in Zambia, and it was just fantastic to see it, because you see the sister tree, and then you see the Suffratex, and amazing. So anyway, we again look at the phylogeny, and we actually saw that there was 30 independent transitions from a Suffratex life form that we could identify, and that was in 26 different genera. And when we looked at when this diversification happened, it was very young. It was 2 to 3 million years ago, so it's very recent. But what's also important that, and it's actually quite logic, because the one is a tree and the one is on the ground, height is a very important character here to distinguish them. But then important also is where they occur, their geographic distribution. It's normally in high rainfall areas, and also where there's great fire frequency. And what we also found looking at this is that our savannas seem to first have to appear here in the tropics, and they became younger as they moved down to the southern African area. And we're actually busy with a paper now looking at hominid evolution using this data set, which is quite exciting. Okay. So something that's very close to our hearts is wildlife crime and endangered species. We do a lot of work on the muti markets, or the medicinal markets. There's several students sitting here that's working on it. This study specifically was done by Janice. Dorcas Lekhaniani is also working quite a bit on the markets. But this forms part of a Global Impact Award program, which is now ended actually, but I hope the DEA will take over the project. So it's a very close collaboration between several institutes in South Africa. The zoo is involved with it, DEA is involved, prosecutors, SIAP. So it's really a big project, but it's also a huge project, a problem in South Africa and globally. But basically what we've tried to do here is to construct a barcoding reference library of all these species that's been traded. And we at ACDB was mostly involved with the plant side, and we did some insect work where Monica and them was doing mammals, SIAP, fish. So it's really a huge collaboration. But one of the big things was the training of researchers. And I remember when Jacques said to me, you have to be trained on how to collect plants. And I said, I've been doing it for 20 years. Now I have to go and do a course on how to collect plants. But it was quite interesting because it has to be under chain of custody so that it actually can be taken to court. So we all learned a lot out of this project. But basically what we wanted to achieve is to use barcoding as evidence in a court of law. So there was a big goal for us there. So one of the nice things that we did is on cycads, encephalatus, which is a big problem in South Africa. We built a nice database, mostly Janice. There's some of the barcoding technicians that worked on it. So at the moment we have 64 of the 65 species that occurs in Africa, and we have 10 or 15 duplicates of most of them. But most importantly, one of these samples is collected under chain of custody. So Jacques should be happy, Francis is happy, we are happy. So for instance, say we get a sample that's confiscated, and we test it and it's Centicosus and it falls into this lineage here, we can actually 100% say this is this species, there's the chain of custody sample, these are the samples that was sequenced using normal barcoding collection techniques. Okay, so that helps a lot. So then Janice also went and she worked a bit on the multi-markets, and this is very alarming because most of the time people think that we're losing cycads just because of the horticultural trade, but actually there is a huge amount of samples in bags on the multi-market. So they're difficult to identify, so because this is what you get on the multi-market, it's no way that you can say it's this species or another species. So Janice actually had to use 5 gene regions to work on this and to sort them out. And it was very alarming to see what actually was on the multi-market. And she just sampled 2 multi-markets, the one in Durban, the Warwick market, and the Faraday multi-market. And I think Natalensis was the only one she found in Durban. The rest was all here in Joburg on the multi-market. And then a very interesting case that we did for the prosecutors is this sample that was confiscated again in Australia. But it had these 13 little seedlings in the parcel, and I'm not sure if I remember correctly, it was something like household goods marked like that. And we had to apply for CITES permits. It came back to UJ or ACDB to be barcoded. In the end, this is what we got from the cycads. Again, near threatened, endangered species, so really a big problem. And I thought it might be just an old lady sending some samples to her son in Australia. It's not the case. So whatever you find, there's always a story behind it. And then something interesting that we did for Francis and Jacques also is these bones that was confiscated. And I think the permit said it was grizzly bears. I can't completely remember. But again, you can't distinguish what is this really. And I think Francis wanted to answer very quickly, and poor Ronnie and Eugene had to work through the night. But in the end, it was lion. So completely something different. So it just shows you the power of DNA barcoding. So. And for those of you that don't know, this is what they use it these days for. So there's a big trade in lion bones. They replace them for tiger bones. So again, huge problem. And something that we've just started on, there's 2 students working on it, Istana and Ryan, is the authentication of herbal products. And this is a huge problem, mislabeling of products. So in this case, we looked at the interchangeable use of Pelargonium pseudoides and reniforms. It's a very important herbal product, not just locally but also overseas, used for cold. We do it in collaboration with Alvaro Fulyun. If you look at, this is now the two species, Sidioides and reniforms, you see the color of the flowers are different, but remember they're not always flowering, and what makes them even more difficult is that the distribution overlaps. So people go and dig them out and they think they have Sidioides and it might be reniforms, or there's only reniforms but they're selling it as Sidioides. You never know whether it was on purpose or not. Also, if you look at the chemical compounds, the Sidioides have this uncalin, this chemical compound, in a very high concentrations, where reniforms, there's very little or none in the compound. So what Ryan did, he bought some products over the internet randomly and tested whether they actually have Sidioides in them. All of them on the label was saying Sidioides. So the results came out, and this is what we got. If you look at the uncalin concentration, there was 3 of the products that had 0 uncalin in. If you look at the barcoding, again, no Sidioides, no Sidioides, no Sidioides. So a case of mislabeling. And this is not just happening in South Africa, it's a huge problem in America, and actually they also now enforcing DNA barcoding to test all the raw products going into the production line. So for us it's good because we will have a job. So one very big project that we are involved in, which I'm very excited about, is barcoding of invasive animal and plant species. And why I like this project is because a lot of what we're doing here is also relevant to our endangered species, trade in endangered species projects. So it's a collaborative project with the DEA, but also with Sajeevan Ratnasingham, who's the developer of the Life Scanner app. And this was a project that was, oh yes, don't forget the barcoding technicians. We have 7 barcoding technicians in the lab that's fully paid by the DEA, so we're very happy for that. But this project was first started just to produce a library for all these species that's invasive in South Africa, but that's also invasive elsewhere in the world. So that was the first step. And then Guy Preston said no, he wants to extend it to the airport. So at the moment, we're just focusing on OR Tambo. It's really in the pilot stage, where we want to include the Life Scanner application and a lab in a box. And I will show you a little bit about the lab in the box. This was launched last year in November during the barcoding conference. And for me, this is one of the big steps that we've taken in barcoding. And so if you look at this, and people that's familiar with a DNA sequencer, it's quite a big instrument. This is where we are now. We have a pocket sequencer, the Oxford Nanopore mini iron sequencer, and this has now allowed us to take the whole ACDB lab and put it in a box like this. This is not brilliant. So what's happening now is officials can go to the airport with a little box, or you can go into the field where you know there's trade in illegal species or endangered species. You can do a quick analysis right there in the field or at the airport, and you can very quickly get an answer of, okay, I have to confiscate this. No, let the guy go. This is not an endangered species. So this is a huge step towards getting barcoding used at the ports of entry in the country. And we busy now, the guy in Canada, Sajiva, is deploying these kits all over the world. Africa, we will in the East. These guys will come to ACDB and we will train them into the use of this. So basically, if we look at this situation at the ports now, where we are at the moment, this step we could never do at the ports, the initial screening and the confiscation. So you know it's an invasive species coming into the country, confiscate it. So now we can do this. There can be a technician at the port doing this. If they have to confiscate, you need some kind of statements. We call it a 212 statement. So the sample is sent to the lab. We do another analysis in the lab, send a report to the agency, and they will then review it and do the prosecution. So for me, this is quite a big step that we're taking. Of course, this is going to take some time to implement it, but I'm very positive about this. And then I showed you this little picture. We now, what next? We want this little barcoder, and there it is. Apparently, according to my Canadian colleague, he said this is going to happen in the next 5 years. He doesn't want to say in the next 2 years, but it's been developed, it's in the testing phase. So we've moved from a whole lab now to this, and all you need is a little pipette and the kit. So for me, this is just wonderful. And your smartphone. At the moment, it's just iPhones that you can use, but apparently they're working on the Android version. And then, again, Prof. Meyer mentioned the PBM project. This is the next phase for DNA barcoding. We want to scale up everything. We are losing species. We can't afford to be asleep. We must just really move. So this project will involve 50 nations. There's going to be 7 biotron nodes where all the sequencing of that continent is going to be happening. I'm happy to report that University of Johannesburg will be one of these nodes. So I'm really looking forward to it. It's a huge amount of money involved in this project. We're still trying to source all this money from our overseas colleagues, but this is just going to be fantastic. And I want to leave you there, acknowledging all these institutions funding us over the past few years. Thank you.
Thank you, Michelle, for that amazing talk, and it's always exciting to see what Michelle has done. Like she has said, I met Michelle when we became partners in the Barcode of Life project. There was a lot of misgivings about the project, but we said we're going to soldier on. And then 1 minute we've started the project with Michelle, the next she's in a 4x4, she's in 4-wheel drive, she's going ahead of us. So it's like we're walking, she's already like at 120 kilometers an hour. So that was amazing to see how much impact she had. So we started the project in 2005, I think. And then all of a sudden she had all these collaborators involved, all of these students involved as well. So I think one of the most amazing things about you, Michelle, is the capacity, the way you bring people in, the way you motivate your students, and the way you get communities or even non-researchers involved into the projects, like Toyota coming on board. She invited me to one of the sampling expeditions, and it was amazing. We had all of these citizen scientists. We had taxonomists, one amazing guy who was very funny, but we had students working interns, and all of these, this sampling process was in a line, like a trend line. Somebody was collecting, somebody was identifying, somebody was photographing. A well-oiled machine would work into the night, would sample with several localities, yeah. So that was amazing, and for me, you were a big inspiration. And I remember one of the first, once we started with the project, she invited me to UJ. She had invited all these African collaborators, and she told me, Monica, you must come and talk about DNA barcoding. I was nervous. I was still doing my PhD, and I was like, I don't think I can do it. She says, no, you can do it. So that was a major change for me because I got involved in a workshop and all of a sudden I was one of the people training people in DNA barcoding. So I think your career, your achievements, the research that she does, beyond adding value to research or to science or changing the current status of research, the impact on government, the impact on applications, and involving government agencies like DEA, she is partnered with them. And all of these changes, innovations, and all of this buy-in from the police is amazing. And I think it's making a big difference in South Africa. In fact, currently, the NZG has got a contract with SAPS, and we do DNA barcoding in police cases. But I think it's the groundwork you laid, the work that you initially did on forensics, on the psychiatrists, even on the Lion-born case. It's convinced our prosecution, our legislative systems. Now we can go into a court, present a case with DNA barcoding. There is no question. The judges believe in DNA barcoding, they believe in the species ID, and we have got strong support. So I congratulate you. I think this promotion was long overdue, and I think there's none such deserving persons like Michelle. So I think she is an asset to this university, and I would like to congratulate you, and a great friend as well. Thank you.
Now, Dr. Mwale, you as well as the Dean, just to come and assist us with the robing of Michelle. Michelle, now you can put on the red gown. Don't know what we're going to do with her, eh? And then the robing, where's the